Okay, this is Introduction to Statistical Learning. We're chapter 11, and we're in the first, I think, of just two questions of the applied questions, all about survival analysis. So here's the questions here, and so as always, we'll just look at them one at a time rather than read them all up front. Well, I guess that's all there is to say about that. Okay, so in order to work through these problems, or through these questions, we'll need to get two packages up, the ISLR2 package that will contain our data, and the survival package that will contain the commands that we will use to execute the survival analysis. So I've already loaded those because it takes a while for those to load. And now once we have ISLR2 up, we can get the data set up, which is called brain cancer, and I'm just calling that D. And we'll see that this is what D looks like, or what the data set looks like, okay, right there. Yeah, I don't have much to say about it. A lot of these things I don't really understand what they are, but luckily we don't need to be medical researchers to do this problem. Okay, so part A is we're going to plot the Kaplan-Meier survival curve with one standard error band from the survfit function, which comes from the survival package. Okay, so first we will fit our survival curve. Is that a coherent sentence? With this command here, and we're calling that fit surv. I might as well just separate these out. How about that? Okay, so we'll run that, and then we will plot it here. Okay, so when we go to plot it, so this wants one standard error. Now normally if I just plot this, there's a command here called confidence intervals that is set to true, and that will plot, I believe, 1.96 standard errors on either side, which encompasses 95%. But it just wants one, not 1.96 or not 2. So we could set this to false, and then manually draw in the lines by plotting against time, plotting this surv, which is the survival line, plus and minus a standard error, one standard error. So I'm going to plot both. The 1.96 or 2 or whatever the default is will be in black, and then the one standard errors will be in red. And this is what we get here. And you'll notice that the red is mostly on the inside until we get to the very limits of the survival function. So the explanation that I just gave to you, I believe is correct, but I'm actually not 100% sure. And this little stuff that's happening at the end gives me pause. So if you have a better explanation, please put it in the description. That would be great. Okay. So now we'll go on to part B. And in part B, we are going to draw bootstrap samples of size 88. Okay, so we're going to draw 200 bootstrap samples, each of them being size 88 from our data. Okay, and then we will compute the resulting Kaplan-Meier survival curve from all of our bootstrap samples. And then we will also estimate the standard error with our bootstrap samples, okay? And then we'll compare those to what we did in part A here. Okay. So the way that I chose to run the bootstrap is the following. So first I set in to in row in D, which is 88, right? There's 88 data points in our original data set. And then I just simply define yi and delta I, which is the things that we're trying to poll, right? Remember, so yi is a particular time of an event, and then delta says whether that event was censoring or, somebody, I guess, dies. And then I'm setting B to 200. Okay, so that's the number of total data sets, bootstrap data sets that we will generate. And then I'm creating this time index here, and I'm sorting it, right? Because in the data set here, the times aren't necessarily in order, so that will just help ease some of the work below. Okay? And then I go through and I build these different matrices, and these matrices will have B columns. Okay, so we'll have 200 columns. So each column will represent a different bootstrapped data set. And then it will have one row for each of the 88 samples. Okay, and then with this big for loop here, we will fill out these matrices one column at a time. And the data that is contained in these matrices will be the things that we need in order to compute, the survival function and the standard error. The survival curve, I should say. Okay, so how does this work? So for each of the 200 samples, sample data sets, okay, for each little b in 1 through big B, the first thing we're going to do is we're going to sample 1 through 88. We're going to get 88 randomly drawn numbers from 1 through 88. And notice that we have replacement equals true, so that means that we could draw certain samples more than once. Okay. And then I'm going to sample yi's. This is just times that something happens. And which ones am I going to sample? Well, I'm going to sample the ones that I just bootstrapped their index number to. Okay. And I will also grab the counterpart delta. Okay. Right. So I'll have a yi and a delta I that both correspond to one another. Again, all based on the boot index. Okay. Now, for each of these columns, again, for each bootstrapped data set, I'm going to go through one cell at a time, one row at a time, and I'm going to evaluate how many surviving members of my cohort that I have. Okay, and that's what's going on here, right? So at each time step, I just sum the number of my data points where something happened after time tj, after a time index. Okay, so I'll fill that out for each of my times, for each of my rows, and then I will also keep track of how many of those were censoring events versus somebody dies with this command here. And then my survival curve is just taking the cumulative product, so the product of each step, of each row and each preceding row by this expression here. The number that are at risk minus the number of events that there were up here, divided by the number that were at risk, just previous to that time point. Right? That's how you generate the survival curve. Okay. So we run that, and now we are meant to plot the survival curve as well as the standard errors. So one way we could do that is so remember what we have here in these matrices, and in particular in boot serve, we have 88 bootstrap samples, each column of which is a different data set. And then each row, if I were to say average over each row, would be the average at each time point, the height of the survival function. Okay, so I would take, if I wanted the average over all bootstrap samples, I would take the row means here. Okay, and if I wanted the standard errors, I can use the supply function, which just takes the standard deviation across each row of boot serve, and this will give me the standard error. Okay? So I can do that. And then we want to compare this to the standard errors that were up here, and I can see that in my confusion here. I've made a little bit of a mistake, but I think it works just fine. I have used 1.96. Okay, so remember up here, we could have set confidence interval to false, in which case it would have just given us the, what the equivalent of these red lines, but we put both in. Here it seems, though, that I didn't put anything in there, but that means that confidence intervals are true, so that I think that is 1.96. And then, for the standard error, 1.96 standard errors on either side of the mean curve, right? So that's from the data. And now we will plot individually or independently the bootstrapped survival curve as well as the standard errors, and I'm using 1.96 instead of 1. Okay, but this again, this should correspond to what gets plotted from the raw data. Okay, so we'll run that. Okay, and it looks like a pretty decent match. The mean curve for sure is dead on, which is very cool to see. I mean, it couldn't be any other way, but it's very cool to see anyway. And the standard errors get a little bit wonky. It seems like everything is shifted down. So I wasn't real satisfied with that. And it occurred to me that possibly what the survival software package is doing is maybe guarding against the possibility that a standard error could be so wide that it actually predicted a negative probability of survival, which is nonsensical. And so I think that they may have done some kind of logging and exponentiation to prevent such a thing from happening. So I also do it that way. So now I just put all of my bootstrap data on the log scale, and and ultimately reverse it. But so you can kind of see how I went about that. So when we do that and we go to plot it, to my eye, this looks quite a bit closer. So that's interesting. You can still see that it's not perfect. Now, some of that misalignment between the two is due to error in the bootstrap sampling process itself. So we can reduce that error by taking more samples, a bigger B. So if I go through and I run that, we'll just run all of them for curiosity's sake. I'm not sure if that one's any better, but when we come here, I think that this one is now better. I mean, it's almost dead on, except for, again, at these extremes. I'm not entirely clear why the extremes get a little bit wonky, but there you go. I'm going to set this back to 200 so that anybody that checks out the Colab sheet will have, what the question asks for. Okay. Okay. In part C, we're meant to fit a Cox proportional hazard that uses all predictors to predict survival. Summarize the findings. Okay, so this is really just 1 or 2 lines of code. So we use this command right here, Cox PH, and then we regress on period. And period means everything that's on the left-hand side of the tilde. Okay, so we run that, and we can get a summary here. It says to summarize the main findings. I never like these questions because the main findings very much depend on the question you asked in the first place, and so that's the appropriate summary. But you can glean what you need to from this probably. Okay. And now we're in part D, which is the final piece here. So we are to first stratify the data by one of the predictor variables called Ki. I'll get to these parentheses here in a second. And then plot the Kaplan-Meier curves for each of the 5 strata, where, again, the 5 strata are based on Ki, adjusted for the other predictors. Okay, and so roughly what we're going to do is we're going to generate, well, I shouldn't say generate, we're going to get the data, we're going to partition the data by its Ki value, and then we're going to apply we're going to apply the model we fit in part C to that subset of data. Yeah, so let's get to these parentheses. It says, since only one observation has ki equals 40, you can group that together with the observation ki equals 60. I decline to do this because, well, for I guess a few reasons. You absolutely could, but my understanding of this variable ki is that it's actually an ordered variable. It's an integer-based scoring system between 0 and 100, I think. And so there is a real sense in which the label 40 means something relative to 30, means something relative to 40, means something relative to 50, and 67, and so on and so forth. And so artificially bumping one of them up seemed maybe not the best thing to do. So there's that. So that also means that when I read the word stratify here, I do not necessarily here break it into groups. What I hear is conditioned on ki. So anyway, okay. So we create our data. Now it also, in order to so we're going to create these data sets and then apply the fitted model to it. Well, you have to define all sorts of different variables here. So I kind of went a little bit further, and I made a different data set for 4 different disease states. Okay. There's one more, which is called NA. And I just always used female as sex, and I used one location. But you could break this down into you could break this down into a lot of different plots, or you could maybe average over male and female, or average over location, etc. So anyway, so we'll go through and make these different data sets, and then we just go through and fit the model we already fit with this new data, these new data sets. Okay. And then we will plot each of these, and then we get our different curves. Okay, so okay, I think that is it for this one.